I don't know what time. I have to look. I think my training's an hour. If y'all want to come after my training.
11:50, I'll be coming right out of the. I just feel bad for.
At what time?
Lunch.
What time?
11:50.
11:50. That should be okay. She has class at 12.
I can text you. I was just wondering because
Oh, yeah. We need to talk about that. Okay, what time? We're ready to go, right? It's time. Who do we have? Nobody's on the Zoom. Okay. We have a few people who are out sick, so we will see. All right, do I have everything? Yes, I believe I do. Right, we did iClicker. So if you are here, please do that. Okay, what slide? Let me see. I gotta find what slides are on. Oh, yay, we have more people. Okay, so we did ABO blood groups, right? And blood group compatibility. Here we are.
So do we want to just not add it's stuff. Or do you want to come in and
Well, how about tomorrow?
That's what I'm just.
Let's do. Yes.
Like I didn't know if we need, I knew we weren't meeting in the lab. I just didn't know if we wanted to come in and talk about.
Well, we could talk about it at 12:50. No, 11:50. 12. Sounds good. Okay. Now ready?
It is fun.
I told him about the heat in here, that it's way too hot. So there's a work order put in. So I know it's hot and we're working on it. I teach in this class every day, so it's worst. I get it.
We had a meeting in here last night.
And it was just hot.
It was terrible.
Then the 104, no, not 104. Biochem one last year.
165.
It's freezing.
I don't understand. I do not understand this building. I don't get it. All right, so what crashes on really small beaches? Microwaves. I giggled. I really giggled at that one, and Charles was like, "Mom, I don't get it." Like, let me explain it to you.
That's even better. Just like when the person's like, I don't know.
I don't get it. Okay, so we're gonna quickly finish our carbohydrate chapter, and then it'll be carbohydrate metabolism, which I think is super interesting too. So we talked last couple of times about how all these different glycoconjugates play roles in cell-cell communication, recognition, cell signaling, exploitation by viruses and bacteria, and all this kind of stuff. So we know that glycoconjugates are super important, right? But you got to think about, we want to try to understand how those connections are happening. What is the sequence of the glycoconjugates in terms of not only the protein portion, but also the sugar portion, right? So trying to understand what's actually happening is the biochemical methods, and we call that glycobiology, right? So there are two big research objectives here, and this is kind of a new field. So what we want to do is, let's say that you isolate a particular glycoprotein, and you want to know, okay, I know this protein is glycosylated. How is it glycosylated? What are the glycan units? How are they sequenced together, right? So that's the first one, identification of structures. And you use liquid chromatography, mass spec, in order to answer those questions. Then you have an array-based method. And this method is really trying to understand binding interactions. The first one is just what is the sequence of sugar, sugar, sugar? Oh, thank you. So the first one is sugar, sugar, sugar. What is it? And then the second one is how are they connected? Right? Y'all do tell me if that ever happens again. Appreciate it.
So like the second one, is what bonds are in between each of the sugar.
Well, like not what bonds, but what portion of the virus hooks on to which receptor in the cells, right? Like a big thing when COVID hit was what cell type is COVID targeting, right? That was a huge thing for people to kind of figure out, okay, now we know where the virus is going. We know what cell type it's attacking. Well, if you understand that, then the next goal is that's going to be the surface protein that we're going to target for antibodies, right? For vaccines, to maybe inhibit the connection between the virus and the cell, right? Maybe we can create an inhibitor. So there are all kinds of targets and things that we can learn in order to treat people, right? Okay, so this is kind of just a pictorial thing of what we just discussed, but you're starting here. You have some kind of cellular glycoconjugate. Maybe it's a protein, maybe it's a lipid, who knows, right? So the question is, what are you going to do? Well, you can go one of two ways. You can go the structural route, where we say, I'm going to isolate these glycoconjugates. I'm going to enzymatically treat them to release just the glycan groups, right? I know what proteins I'm isolating, but what are the glycan groups on those proteins? And you can go through liquid chromatography and you can identify what the groups are, but this gets a little tricky. And the reason it gets tricky is because unlike like DNA, you only have 4 base pairs, right? How many different glycan groups do we have? A lot, right? And not only do you have a lot of glycan groups, but you can also have stereoisomers of those sugars. So we're talking about a huge degree of variation that could occur. So yes, this is possible, but is it going to answer all of our questions? No, but nothing in science ever does, right? But that's okay because we're still learning stuff. You can also go to mass spec, right? Mass spec is just going to kind of tell you which glycan groups are common, right? So you can look at a subset and you can say, oh, well, this is the most common glycan group that we see on this set of proteins, rather than sequence and cut it one at a time and say, okay, it's glucose, N-acetylglucosamine, right? That's what you're doing up here with liquid chromatography. Down here, you're saying which of the groups, right? You're looking at kind of as a whole. Okay.
So
We're going to talk about that.
okay.
We're going to talk about
You're
that.
seeing where my head is.
I know exactly where you're going, and we're actually going to talk about that. Yes. Okay. So the other way that you can do this, right, is if you isolate these glycoconjugates, you can do a couple of different things with them. You can actually create an array of lectins, right? What's a lectin? It's something that, it's a protein that binds sugars. Any protein that binds sugars. Not all, okay, all lectins bind sugars. Not all things that bind sugars are lectins. So I just blew, you got me? So say that one more time. All lectins bind sugars. Not everything that binds sugars is a lectin. Yeah.
So lectins are specifically proteins, or
Yes, lectins are specifically proteins.
so basically the alga comes in where it's any protein.
It could be a protein, it could be a non-protein. There are many things that bind sugars.
No, but as a lectin.
All lectins are proteins that bind sugars.
Okay.
All right, so you can create an array. Do you remember when we talked about those mRNA arrays and things like that, right? Back in Biochem 1, you attach this protein to the surface of a slide or anything, right? Now, if I label these glycoconjugates, I can actually see which ones bind to which lectins and use fluorescence, right, as a readout. Okay, I have these, they'll bind to these lectins. Well, I know that this lectin is on this bacteria, so maybe that's how it's happening, right? The other way that you can do this is you can actually bind the glycoprotein itself or an antibody to the glycoprotein. Either way will get the glycoprotein to attach. And then you can add other stuff on top of it. So like I could put the glycoconjugate on the slide and then in reverse label the lectin. Or I could even take a whole virus, label the outside of the virus, incubate the virus with this array of glycoconjugates and see which one it adheres to, right? So there's a lot of different ways that you can do this array stuff. Okay, so let's talk about details, right? So if you want to do the structural route, right, we're gonna first talk about this side of the diagram, then we're gonna talk about this side. So we're gonna go to the structural route first. In order to understand the structure of your glycan groups, the first thing that you have to do is separate it, right? So a lot of them can be embedded in the membrane. So a lot of times you have to treat it with some kind of a detergent that will break up the cell membrane. Depends on where it is, what you do to release it, right? But you basically purify your glycoprotein. So now I know I have a protein that's attached to a glycan group, but there are two different ways that proteins are attached to glycan groups, right? It can be either, as you were saying, which one is N-linked? Asparagine is N-linked, and serine threonine is O-linked. So first thing I want to do is I want to cut the sugar off of the protein, but I don't want to cut my sugar linkages. So the easiest one is my N-linked. If I have a specific enzyme, which of course we do because we have so many specific enzymes, that will cleave N linkages, PNGase F. This enzyme will cleave only N linkages. So I can separate my N-linked glycan from the protein. So that's easy, right? So now I can have a way to separate my N-linked things from my O-linked. That's great, right? So that was a win-win. Now I have a serine threonine. I can actually do a chemical treatment with sodium hydroxide, and I can cleave off my O-linked from the protein, but it won't cleave the sugar. So you could do this chemically. You could actually do it enzymatically as well, but it's a heck of a lot cheaper to do it this way. The only reason why this is a chemical and that's an enzyme. Okay, so now what do? Now we want to identify piece by piece which sugars are attached, right? So do you remember how I said that there were enzymes that catalyze the reversible reaction of adding a sugar group to the chain? And that each addition was a particular enzyme that did that addition? Okay, so like if, I don't remember which one, green is glucose, right? I don't remember the color scheme, but like if green is glucose and blue squares are galactose and yellow triangles are N-glucosamine, right? Each one's different. So there's one particular enzyme that will look at an N-acetylglucosamine and be able to add an N-acetylgalactosamine to it. But it is one enzyme. And that enzyme is perfectly specific to add the one type of sugar to the N-acetylglucosamine. And it has to be an N-acetylgalactosamine. Highly, highly specific. But notice I said a reversible reaction. So what can I do now with those enzymes? I can one at a time run them in the opposite direction. And what will happen? Piece by piece, I will break down my glycan groups. So if I treat with enzyme A, only things that are attached where I have an N-acetylgalactosamine connected to an N-acetylglucosamine will actually be cleaved. So I can add these in sequence and then analyze them by liquid chromatography. And depending on where the peak is, where it elutes, right, in time will tell me how big the moiety is, and the enzyme will tell me which moiety comes off. And from size and knowing which enzyme is cleaving, I can figure out exactly which glycan groups were there and in what order. Isn't that cool? So by using the enzymes in sequence and analyzing the size product that comes out, I can tell which residues were there, which glycan groups were there, and how they were arranged, right? Now, you might not know if it's an isomer, right? You might not know that. But you have a lot more information than before. Absolutely. Does that make sense? So you can also determine overall, if you look at a whole glycan group, right? You can run a mixture of proteins, right, through mass spec. You take these proteins that are glycosylated, you cut off the glycan groups, you run those glycan groups through mass spec, and now I can say, well, we have a bunch of different glycosylations going on, right? So this set of proteins isn't just one glycosyl group, right? We have kind of a range of glycosyl groups, and we can identify based on molecular weight which ones are which. So there are multiple ways to kind of gain information about those glycan groups.
Can you make sense of that first bullet point? Basically what it determines it.
So you can use, so do you remember when we talked about proteins being really too big, right, to run through? So you can actually cleave it the same way you do a protein with an enzyme that we were talking about before to make them smaller if you need to. So you can, the smaller it is, the better the ionization efficiency is in mass spec. So if you break them down a little, you'll get more ionization, but then you have to be able to backtrack. So
And that's why they just the glycogen group rather than
the protein, okay, you'd never get any information. Be too much. You wouldn't be able to do it. All right, so our high throughput arrays, these are really cool. You can bind different things. You can bind whatever you want to an array. That's the really cool part of an array. You can put whatever you want, but you have to remember that if you are building the array, what are you automatically putting into that system? A bias, right? Do you remember when we talked about the RNA-seq and then the RNA arrays, and how the sequence one, there was no bias because you sequence everything, every single mRNA that was there, you sequence it and you figure out which transcripts are there. But the array, you start with a grid, you pre-built the grid. You decided what kind of DNA was on that grid. And therefore the only things that bind would be things that you already thought were there, right? So you have an inherent bias. Anytime you use array things like this, you have to remember that you have bias. So you can use it though, not only for plus or minus binding, you can also use it as a quantification, a level of binding, because the more binding you have, the higher the fluorescence is going to be. Right, and you can also do different washes to see how tight that binding is. So you can get a little bit more of an affinity. So here's our examples, right? So this one's really, really common, a lectin array. If you start with a lectin array, you are actually adhering the protein to the glass slide or whatever it is, right? So L1, L2, L3, L4, maybe these are lectins that are found on different cell types, right? Maybe this is lung epithelial cells. Maybe this is nasal epithelial cells, right? So maybe we track the path of how a pathogen gets to your lungs, and we try surface proteins that are lectins along that path and see, okay, what is COVID going to bind to? So you take the virus and you label the virus with some kind of fluorescent probe. It's relatively easy to do. And then you take this virus and you put it all over this glass cover slip, whatever it is, and then you wash it. And when you look at the readout, depending on which circle has which lectin in it, right, will tell you where it's binding. But do you see how they have some that are super dark? There are some like, oh, maybe I could kind of see a little something, right? You can actually correlate intensity of fluorescence with binding affinity. It's not a perfect, like it's not the optimal way to determine binding affinity, but it does work. It does work. They've done a lot of this with bacteria. The other thing is it doesn't necessarily have to be something bad. It doesn't have to be a virus or a bacteria. Sometimes we want to understand how you have cell-cell communication, right? How does that white blood cell stick to the epithelial cell of the vein? Well, then I'm going to look for, okay, I know there's glycoproteins on the outside of a white blood cell, and here are all the different lectins that are on the outside of an epithelial cell, a vein epithelial cell. So now I'm going to try every one of those lectins. Which one is it binding to? So either way, you're going to gain information about how the system works. And the other way we said earlier is you can do this with an antibody system where you want to put the glycoprotein on the slide itself, right? So there are two different ways that you could do this. This is what we were kind of talking about earlier. If you have the antibody, we know the antibody works to recognize this protein that's glycosylated. But then I have this range of either glycosylated glycoproteins or lectins, and we look for what interaction occurs. It's very similar to doing it this way, except instead of adhering the lectin to the array, we're going to adhere basically the protein via an antibody. But it's the two are very similar. The other thing that you can do is you can say, okay, I'm looking for a particular glycosylated protein. I know that this lectin recognizes this particular sequence of sugars. So I want to know which glycosylated proteins have that particular sequence of sugars. And so you can attach an antibody that recognizes that sugar sequence and select out those glycoproteins that have the sequence you're looking for. There's a lot of different ways that you can gain information about glycosylated proteins. And this is really new. It's really something we haven't thought about before, right? You think, okay, well, how many proteins do we have in the cell? Well, you can get a number on that, right? But there's an added layer of diversity. So this is what you're going to learn as you learn more about how your whole body works, how the cell works, right? Well, we think, oh, that's pretty diverse, right? You have 20,000 different genes, right? So each gene can make a protein. Well, not quite, right? Because you can have different splicing. So that gives you a bigger variety. So once you have those different splice variants, now maybe there are different post-translational modifications that occur to those proteins. One of them being glycosylation. So how many different glycosylations can you have? Well, maybe if the protein's glycosylated this way, it behaves X. And if it's glycosylated a different way, it behaves Y. So what you thought was a simple soup of proteins is not. And that's a very small portion of the diversity that, that is required to make your cells work. Well, it's like every time we learn something, we're like, oh, it's way more complex than we thought. Yeah, that's how science works. That's how science works. All right. So I think that is, so that's the other thing. I forgot about this. You can skip the antibody altogether and you can actually attach the proteins or the sugar groups to the slide itself. So you can skip the antibody if you want. And I think that's what we showed in that very first slide. I don't remember. It's all kind of different ones, but they work the same way, right? You attach something to the slide, you radio label whatever you're adding, and you see how much and where it lights up. All right. Okay, that's it. That's all I got. Questions?
You did not answer the question
I didn't. Okay.
because it was something completely different and it was kind of more just me wondering.
Okay.
So serine and threonine have the linkages and
Yes.
asparagine has linkages.
Yes.
Slide before that. What does methionine have?
What does methionine have?
First thought,
What?
my first thought was sulfur linkage. But I didn't know for sure.
Yes. It's not very common, but it can.
Okay, but that I was just like, no.
No, not necessarily. It's way more uncommon. Most of the time, you're going to see serine, threonine, O linkages, and asparagine N linkages.
Okay.
That's the the bulk of it. Okay. Good. Questions? And y'all know we have an exam coming up. Coming up.
After school goes Monday.
Is it after?
Yeah, so not next week. Definitely, yeah.
So what is it? Let's see. So let's see. I know. I gotta make sure. Is it really?
Yep.
That's so nice, right? Let's see what day it is.
The 17th.
The 17th. I always like to look. Which one? No.
Yeah, it's on that Thursday.
Here. Class calendar. I'm always like, where did I put it? Yeah. So 17. That's right. 17. So we have. So all we're doing is talking about more carbs, and then exam one.
But yeah, it's on the same day as the sugar. So,
It is what it is. Start studying. Start studying.
back to back. Like back to back.
Okay. So I am gonna leave up y'all stuff and let y'all come and take the floor. Yeah, you can write all over the one you keep. So everybody's turned in their papers, right? I need everybody's papers. And then I'm gonna come be a student. Y'all have y'all papers. You haven't given them yet? No, but I want you to have it like, as we're talking about this, if we're gonna be like, oh, did they say that? Oh, did they say the patient did this? Or, you know, like, we're gonna be talking about it, you're gonna want it. Yeah, you're gonna want it. I guess I should have said you could have had it electronic. Right? Okay, anybody else? Good? All right.
Dr. B, can I pull my mask down while I talk, or do you prefer that I keep it
Yeah,
on?
I think if you're behind the shield while you're presenting.
Okay.
I think
I'm-
you take off, y'all, is everybody okay with that?
I've been tested repeatedly,
I know,
so
I know it's
we all
hard. It's hard
good.
to be the presenter and have the
Mhm.
mask, so I get it.
Okay.
Everybody came with their stuff. I want one. All right, so good to go.
Ready? Today we'll be presenting a case study on simple sugars far and wide, yet not a grain to be metabolized. And it's by me, Madison Orlando, and then Brooke Bergeron, Alan Correa, and Tyler Verrett. So a patient saw her primary care physician. She's a 57-year-old woman. She's experiencing nausea and vomiting for the past 5 days. She was unable to lay flat without becoming breathless. And when she was with her primary care physician, her temperature was 104 or 100.4 Fahrenheit, and her oxygen saturation was at 85%. Oh,
So what do y'all think about those things?
can I move this, Dr. B?
Oh, drag it up. So click on the back or drag it down, whichever one works better. And it'll disappear. After a second, it'll disappear.
Okay.
It will. Yeah. So extreme conditions, like should she be in the ER? Yes, absolutely. Yeah. This patient.
Just based off of her pre-existing conditions.
Oh my gosh.
Wait till y'all see her list of medicine. She's got quite a cocktail going.
Oh, that was just me last week.
Yeah.
Yeah, no, my thing was she experienced nausea and vomiting for the past 5 days. If you have vomiting for 5 days,
Yeah.
That's,
And that O2 is low.
and that O2 stat is so low.
Less than 90.
Yeah.
Yeah, like that's probably where we really.
So I think it's really important as we go through these, to highlight the things that are like, holy crap. You know, just so that you're starting to recognize the things that are critically important.
I want to know how long she was like, I can handle it. I can handle
So
it.
actually, once I started to research, women are statistically less likely to seek care because I do the same thing. I'm like, it's fine. Like, it's not a big deal. And then it is a big deal.
You know,
Yeah.
And the doctor's like, oh, no, seriously?
That's-
Like,
yeah.
Good.
Yeah.
I'm like, oh, no.
I'm out.
You should.
I mean, if it was this, I would go to a doctor.
Oh, yeah.
But normally I would not.
Definitely. Because so her pre-existing condition, she was diagnosed 25 years ago with insulin-dependent diabetes, and that's going to be type 1 diabetes. She has a coronary artery disease. She has past myocardial infarction, which is just a heart attack. She has end-stage kidney disease requiring CAPD, autonomic neuropathy with orthostatic hypotension, and gastroparesis.
So let's talk about what those things kind of are, right? So we talked about diabetes, right? Coronary artery disease. What is coronary? It's your heart. So these are the blood vessels that feed your heart. Okay. Past myocardial, y'all said heart attack.
Mhm.
End stage kidney disease requiring CAPD. What is CAPD? Dialysis.
Mhm.
So there are 2 types of dialysis, right? So you can have hemodialysis, just hemodialysis. I'm like, there's too many types of the term. Where on one of your arms, you have basically something going from a vein to an artery, right? So you pull out the dirty blood, right? And then it comes back in clean, right? It goes to a dialysis machine, it cleans it out, puts it back in. That's hemodialysis. But peritoneal is where what they do is they fill your belly with fluid. And just by diffusion, all of the dirty stuff that's in your blood, because you have lots of small blood vessels in your abdomen, will actually diffuse into the peritoneal fluid that you filled it with, and then you just drain it out. So usually you do that for longer, like a whole 8 hours, 10 hours. So when you go to sleep, you do it like sleep. Whereas hemodialysis, you do usually for a shorter amount of time. But if you do it for a longer duration at a lower pressure, it's much better for your body. So if you know that you're going to be on it long term, you want to do it as slow as possible. But you can also do it. Yeah.
What did you say was the hemodialysis? One arm?
In one arm, you have basically a, oh gosh, what do they call that? A shunt. Yeah, a shunt. And you're gonna pull blood out that usually goes through the kidneys, right? So you're gonna pull it out, you're gonna go through the dialysis machine, and it's simple diffusion where you have all of the things that your kidneys normally filter out, the machine will filter out, and then you put it back in.
Is that one less common?
No, not necessarily. You'll see a lot of people who are on dialysis. You'll see them wear long sleeves because it actually makes the vessels inside of your arm balloon up, and so like they don't want people to see that. So a lot of times those people wear long sleeves. So you just don't see it.
Answer questions throughout the study or more.
Well, like if it's appropriate, if y'all are okay with it,
Yeah.
I Yeah.
laid back.
Yeah, I mean, I
Because-
don't expect it to be like stiff, whatever,
Okay,
you know.
'cause
And
I
we have
guess-
the whole class period, so it's not like
Yeah.
we're in a really big rush. Yeah, fish filler. Yeah. Yeah. So, yeah, if anybody has anything to contribute, please. Do they do? See, I haven't seen anybody with that. Yeah. Oh my
Okay, real quick. So we have,
So we said autonomic neuropathy, which is, anybody can contribute, anybody. Yeah, nerve therapy. Autonomic meaning, yeah, all of your like breathing, heart rate, things that you don't think about.
Is low blood pressure.
mhm. Yes.
And then gastroparesis. Anybody? What is that? Less slow digestion. Yeah, like takes longer.
We all think that, it was just for diabetes that caused all the other things.
I think it definitely played a part in it, because once we get further into what's actually going on, like her symptoms, because this is just at her primary care physician, we didn't even get to her hospital,
Oh, God.
like admission, I guess. Yeah,
Yeah.
she has a lot going
Yeah,
on.
so they'll make us talk.
Yeah. There's
Yeah, 'cause
a
that was my
lot.
thing, was the diabetes, like, almost like
It's-
a predisposition.
Once we get to I think the edema kind of set off like a red flag.
All right,
So,
keep going.
okay, so these are her relevant exams and labs. This is still with her primary care physician, so we haven't even made it to the hospital yet. So they did a BQ scan, which did not show evidence of a pulmonary embolus.
So what is that?
It's the blood clot.
Yeah, blood clot. So there's no, if there's fluid in the lungs, well, no, if the lungs are not, if she's out of breath, right?
Mhm.
If her lungs are not functioning.
Yeah.
The question is, why are her lungs not functioning? If they're not getting blood, they're not functioning, but that's not the problem.
And then her admission lab results, so this is when she gets to the hospital. She has elevated creatine kinase and troponin 1 levels. And the elevated levels were in line with a myocardial injury, so she was admitted to the CCU.
Right. So what are creatine kinase and troponin 1? We're talking about a lot. So they are cardiac proteins. These are proteins that are supposed to be inside of a cardiac cell. Where are they? In her blood.
Yeah, shoot, no,
Which
that's
means
like,
that the cells have broken open because they have died and it is not good.
I'll go next.
Click that, there should be a forward backwards maybe.
Doing the side, the up, the down.
It's okay, I like the stretching spot.
Oh,
Just click it.
I just clicked and then I use the roller. I use the roller. So try that.
Oh,
For future.
Okay, so what is diabetes mellitus? Mellitus, yes. It's just refers to diabetes. We have type 1 and type 2, and it kind of describes how the body processes glucose. So after you eat carbohydrates, the body turns the carbs into glucose, which is a sugar compound that's released into the bloodstream. And in order to remove the glucose from the bloodstream, you're going to need insulin, which is secreted from the pancreas. So in the case of our patient, she has type 1 diabetes. Her immune system attacks the cells in her pancreas that secrete the insulin. So there is no insulin to remove the glucose from her bloodstream. So if it's left untreated, it can lead to heart disease, nerve damage, blindness, and amputation of the limbs. But she's already showing signs of the first two, so you know it's severe. And she's been diagnosed for 25 years now, so it's not, you know, a new diagnosis. And what does the insulin dependence suggest about the type of diabetes that this patient suffers from? She's insulin dependent, so she does have type 1.
So what's the type 1 and type 2?
Type 1 is going to be, you know, your autoimmune disease.
Don't make insulin.
Yeah.
Therefore, you are now dependent upon an external source
Yes.
of insulin.
And then type 2 is going to be lifestyle based.
Well, yeah, so anybody, what's type 2?
Like that.
They're insulin resistant, so their body makes insulin, but their cells don't respond to it. So you can control usually type 2 with diet, exercise, right? Limit the amount of sugar that you consume so that basically you're making insulin less often. Your body responds to it more effectively. The type 1, you just don't have it.
Yeah.
So you have to. You have to. But you can progress from type 2 to type 1. It happens.
Progress from type 2 to type 1?
Type 2 to type 1. Because if your cells keep overproducing insulin, like trying to get your cells to respond and keep making insulin, like it wears those cells out. So then they stop making insulin if you turn from a type 2 to a type 1.
Okay, what's up?
So it's like a general stuff.
Uh-huh.
How would you say that insulin regulates the blood flow?
Well, we'll get to that. We'll get
I'm
to
actually
that.
about to,
Yeah.
Yeah.
Oh, I'm sorry.
Keep on, no, no.
so, have it a little bit further down, but, let's see, we'll go to the next one. So why did this patient not complain of chest pain? One of the most common symptoms of a heart attack. So like I said, women are a lot less likely to seek care, and actually women tend to experience a little bit more of like the odd symptoms associated with heart attacks. So that can include like jaw pain, heartburn, you know, stuff that's not necessarily chest pain, you know, the usual symptoms that you would see. And one thing that I found during research is actually creatine kinase is not kind of like the marker that is typically used now it's not like the big red flag where it's like, okay, this patient, you know, may have had a heart attack. It's more of the troponin 1 that becomes, you know, the bigger red flag. So which they are both present, both are elevated. So then you could go ahead and assume that it's a, you know, some type of heart related issue. But if you just have elevated creatine kinase, it can even be linked to excessive, like long-term exercise. So it's a little bit less of a specific marker. So as of right now, before we've read like any further into the case study, we don't know if it's actually a heart attack. It's just a kind of heart-related injury.
So you're saying that, like, as of right now,
Uh-huh,
obviously, that it may not have been a myocardial infarction.
Uh-huh.
But we also know that she suffers from autonomic neuropathy.
Mhm.
So is there a way that that could be playing a factor into why she's not complaining of chest pain?
Don't know because they didn't specify like what it was related to. I don't really like the description that they gave of her admission symptoms because it's very vague, and then they don't follow up with all of her information once she gets to the hospital. So like her temperature was updated, I think her blood pressure was updated, but some stuff it was just left with her primary care
Yeah,
physician.
if it hadn't changed, it's
So,
probably whatever it was when she was
yeah.
at the primary care. So unless they say different, like her temperature spiked to 104,
Yeah.
you know.
And
something.
her temperature did eventually go down once she got to the hospital, because I think the next slide is going to be her a little bit deeper into her admission symptoms.
But with neuropathy would be like a localized thing, so you could say you have neuropathy like under
Mhm.
your arm, like if they were more specific, that would
Mhm.
tell you if that's why the chest pain is
Yeah.
different.
Yeah, definitely.
Yeah, well, she's already had a heart attack. I mean,
Oh yeah,
she's
she
already
has
had
a
heart
history.
attack. So.
And you would think she would know or be
But it could be
familiar,
different.
and it can.
Every time.
And the symptoms are so weird. It was a little scary when I was reading it. Like,
Yeah, the woman knows she's a little bit.
yeah.
All right, keep going.
Okay. So this is her
That's a lot.
Okay, we're not gonna talk
Yeah,
about
no,
every single
but just
medicine,
the list.
but this is organized very nicely. I love this.
It's a lot. It's a lot.
It's just like-
So she's taking like 20 pills a day on top of her insulin. So
Like this is.
we got some issues.
Oh, this is when my grandmother got old, we would go over and do pills and bills. Pills and
Oh,
pills and bills.
okay.
All
All right, so this is where we get to her physical exam. It's a little bit more specific than her primary care physician information. So her temperature did drop. Her blood pressure was 177/90. Heart rate was 103 beats per minute. We have a respiratory rate of 22 per minute. She is drowsy, but she is alert once stimulated. Her pupils were fine, equal, round, responsive to light. The whites of her eyes were not yellowed. She had one healed carotid endarterectomy scar on the left side of her neck. So, yeah. So we've had some surgeries. And then they were within normal volume range and there were no fruits present. And
But what's really concerning is what? Look at that
The
blood
blood
pressure.
pressure.
No,
Really
look at
high.
that number.
Because isn't the normal blood pressure like 120/80?
Yes,
Mhm.
Yes, that is.
that's really high. That's really,
so
really high.
temperature went down, but then blood pressure kind of got some issues.
Hey, yo.
And I do apologize if I butcher these words.
You're fine. It's fine.
It's, yeah,
It's
so-
lots of information.
it was a lot. I tried to read like beforehand to try to kind of familiarize, but it didn't stick. So we have the patient's jugular venous pressure was elevated at around 12 centimeters of water and her lungs were dull at bilateral bases with rales 1/4 of the way up. A heart exam showed a diffuse and laterally placed PMI, which suggested a cardiac enlargement.
So what are those things, let's talk about that real quick. What is in the lungs, bilateral bases with rails, 1/4 of the way up? What is that telling us? Right now? Okay, look at what that. What's her lung sound? That's lung sound, those are lung sounds. So if you have fluid in the lungs, you hear rails as like crackles and things like that,
Mhm.
right? So you hear them like sticking to each other and coming apart, and so it sounds different,
Mhm.
right? So you have fluid inside the lungs.
Yeah.
That's what sounds.
The little crackling sound, they let me hear it too, and I can hear it there. That's when it's disgusting. It's like fluid in the lungs.
Disgusting.
Yeah.
Body.
Yeah, so we know something's up with the lungs, right?
Mhm.
Okay, and then the heart exam, which is diffuse and laterally just like PMI. What is that? Ready?
I know it's not necessarily like.
What does it suggest?
Cardiac enlargement.
Cardiac enlargement.
So swollen tissue.
So basically what's happening here is that you have portions of the heart that are rubbing in places where they shouldn't be rubbing. Yeah. So, and usually that means that the heart has expanded more than it should. And we'll talk about why. Okay.
So despite her previous exams, her heart rate was regular with a normal S1, S2, and S4 gallop that was consistent with reduced compliance of the left ventricle.
So where does the left ventricle pump to? To the aorta, to the whole body. Left goes to the body, right goes to the lungs.
Yes.
What did you say? You said like the lower what was that? Yeah, like the left side.
Oh, so the right side goes to the aorta, goes to the body. So it comes into the heart, goes to the right side to lungs, lungs to left, left to body. So if your left side fails, what you're gonna have is back up into the lungs because you're not getting it out to the body. Yeah, so the blood is gonna pool in the lungs. If the blood pools in the lungs, it's gonna have more time to diffuse into the pleural space into all those spaces where you're not supposed to have fluid, and you're gonna have fluid on your lungs.
Can I add to that? So like whenever
Please do.
it had like the jugular venous pressure and stuff, that was showing that there was too much pressure on the right side. Because like think of how your heart is and stuff. So like on the right side, it's gonna be coming from like if the blood's gonna be backing up, it's gonna kind of come up right here. So that's why you kind of tell to have like that bigger venous pressure right here. And then like kind of shows like the whole heart pretty much like overworked.
So when you're looking at a patient, right, it's really important to look at all the different little pieces.
It's a
It's like
lot.
the same puzzle.
It's a lot.
That you have to put it all together, right? It doesn't, each individual thing could mean something different. But when you
And she
put it
has
all
so
together-
many pre-existing conditions on its own. It was
Also, if,
It's a lot.
muscle, like that, it's gonna take a bigger, kind of like it's muscle that was, so big,
Yeah, because you're still trying to pump out the same amount of blood to the body. So what's gonna happen is the heart's gonna enlarge so that each pump has to get bigger in order to send out the same amount of pumps. Oh yeah, it's trying hard. It's trying hard. It really is trying.
Also, that kind of work is changing a lot too, because the bigger the left ventricle is, the less room you have for blood, less you should have to punch. So she passed.
I kind of like thinking like all the muscles in your body, so like the more they work, they kind of get bigger. And so like your heart's kind of the same thing. So the more each individual kind of works and stuff, it's gonna get bigger, bigger, bigger. But your heart has to be a big.
And it's gonna get all that damage from there.
okay. So last of her physical exam, we have no murmurs or rubs present. Her bowel sounds were normal. She does have one catheter present in the left quadrant, and we're going to go ahead and assume that's her dialysis port. And the site was clean, dry, and in good condition. Her abdomen was soft and tender. No signs of hepatosplenomegaly.
So hepato referring to
Liver,
liver,
uh-huh.
spleen, and megaly meaning enlarged. So
Okay.
usually, there are indications, like if you have an infection, your spleen is gonna be enlarged, you know? There are certain things that both that is indicated.
So she was alert. She knew date, time, place, no obvious motor or sensory effects. She did have a positive one pedal edema present. So edema is going to be the swelling of your tissues when excess fluid is present. And a +1 is not terrible. The scale is 1 to 4. So 4 is going to be your worst. You're going to have a super deep indent if you were like to press her skin and it wouldn't disappear right away. But hers, it wasn't very deep and went away very quickly. But we have some pooling of liquid going on near the bottom half of her body. So what are some typical indicators of cardiac injury? The most typical are going to be chest pain, shortness of breath for men.
For men, it's chest pain.
Yes. So we're going to have shortness of breath, fatigue for days or weeks before the injury, nausea, faintness, sudden sweating, abnormal heartbeat, blue tint to the lips, hands or feet, and loss of consciousness. However, in women, you see much more subtle and different effects. Because like if I would have heartburn right now, which I do get heartburn frequently now, like I wouldn't assume a heart attack, like
No, my grandmother said.
At all.
I just felt like my bra was too tight. You know?
yeah,
Like,
it's scary. Very vague. So that's why we see women, people of color, elderly patients, and patients with past heart failure, diabetes, or stroke may not show any or very, very mild
So
symptoms.
she's already getting older. She's got diabetes. She's had a heart failure. I mean,
She's a woman.
she's
We've got,
a woman. We're taking up all the things.
she
that are
meets every
non-traditional.
criteria, essentially.
Yeah.
And is edema a common complication with diabetes, and does the +1 grade indicate severe edema? So we know her edema is not severe. The +1 grade is probably the lowest grade on the common scale that is used to assess edema. But edema is a term used to describe excess fluid buildup in parts of the body, but is particularly seen in the hands, arms, feet, ankles, and legs. So the fluid builds up in small blood vessels, causing swelling, and is commonly seen in diabetes patients. And diabetes patients also have issues with blood flow, so this makes edema especially complicated. And my uncle is actually a radiology tech in a cath lab at CIS.
Nice.
So he took me to work with him one day. He just brought me to work with him. But they had introduced new technology that like drastically reduced amputations with diabetes patients. And I got to see the procedure like through the little like sitting area. So they use x-rays and they go in with stents through the veins and open the veins up. And yeah, so they drastically improved like the, I guess,
The outcome.
yes. So
Yeah,
very cool.
that's awesome.
We'll switch over.
Okay, so now we get into her lab results. So first, her creatine kinase was high. I put the normal level up there, which it actually wasn't high in an overall level, but there's 3 fractions of creatine kinase. You have MM, which is for the body and the muscles. You have MB, which is the heart fraction, and BB, which is actually the brain fraction. And her MB fraction is what was high. So that goes back to like all of the heart problems we were having before, saying that something's definitely wrong with her heart. So troponin 1 is another indicator of something wrong with her heart. Her level is quite high for troponin 1. And that one is, it's not the one that spikes, it's not the one that spikes immediately, but it's a little gradually after. So it's usually the one that catches heart attacks. I should have put the chart up with all the,
Well, yeah, so y'all remember
The-
we have the lab, and there was when we were purifying LPH. LPH is another one of those proteins. LPH is way later, right? But that each one spikes at a different time, and you take a blood sample and you look at where it's spiking, and you can see how long it's been since they got on.
the troponin 1 spikes around 6 hours after, or 12, 6 to 12. It kind of varies depending on the person, and it stays high for a couple weeks. And then, so her blood glucose was also really high. And being that she's on insulin and already on a regimen, this is really concerning because it means that it's not well controlled. And when your blood glucose gets uncontrolled, you get hyperglycemia, which causes all of those things. And that's honestly probably why she has half the diseases that she does have with her kidneys, with her lungs and her heart, because that hyperglycemia causes a lot of issues within the body.
So if you have diabetes, that you control it, like, you know, someone who has diabetes, you know, they're like, oh, it's not a big deal, I'm gonna have, no. Let's take it
Very
there.
serious.
Yeah, help them.
Yeah. And so actually the recommendations for people with diabetes is a little higher because obviously their blood glucose will be slightly higher, but hers was excessively high at 286, which is way above.
Was this fasting or was this 2 hours after a meal?
It didn't
Doesn't
really
matter.
say, but either way, yeah,
No,
it's
I know
still
it's
really
bad.
high.
She probably wasn't eating because of their vomiting.
Right.
So
that's,
you know, I doubt she's eating a lot.
okay. And so coronary angiography is the X-ray imaging of the heart. So it'll look like that. And I don't think that that one has any, blockages. So the issue with going back to like how she had the venous pressure up here. So if her heart arteries are blocked, the ones going up, if they're blocked, that will increase her blood pressure as well. If there's any plaques in your arteries, it's like, I think we did this in physics. When you move something faster through a smaller hole, the pressure is going to be higher. So that any plaques in arteries anywhere in the body increases your blood pressure. And that honestly is a big issue with like the limbs and things like that. If you get plaques in your legs, that's when your legs typically get amputated, things like that. I think that's it.
What is collateral circulation? How does it help with recovery? Well, basically, the body responds to the lack of blood flow to the tissues. So it's kind of like a highway, you're building extra little roads and all around the tissue in order to provide the necessary blood. So it happens during true endogenous tissues like the heart. It also protects against ischemic injury, that may be caused by thrombus, emboli, calcified plaque, all those things. What is collateral circulation good for? It helps reduce ischemia, more blood, less stress in the heart, less ischemia, preserves ventricular function, which means that the left ventricle will be able to have more blood, more oxygen to do its functions, and has better prognosis. The patient will have more years, of
Right,
healthy
because a
quality
lot
life.
of times when you have like some huge blockage like that, right? What they want to do is they want to do a bypass surgery, but were they able to do that on this patient? No. So they looked and they said, okay, she's got collateral vessels. So, well, I'll do her for a little while.
So what is atherosclerosis? It's basically an inflammatory disease that's caused by the accumulation of plaque around the genius tissues of the vessels, mainly the coronary arteries, the arteries. It can cause things like ischemic strokes, atherosclerosis, blockages, myocardial infarctions. A good collateral situation is associated with a lower Q wave incidences. Q wave is basically one of the letters that we use, the electrocardiogram,
Did y'all have a picture? Don't put it in. Okay, that's okay. That's good. But so you look at the electricity of the heart with the EKG, and so you can tell when the cells, in order to contract, they have to go through a polarization and a depolarization, right? To contract, relax, contract, relax. And so you can actually look at the electricity that the heart gives off when those polarization, depolarization events occur. So you have small polarization, depolarization. Look at the atria, right? At the top of the heart, right? But when you look at the ventricles, it's a much bigger electrical pulse because it's a bigger part of the heart. Oh, she's right. Can you label it PQRS? Yes. Yay! Well, so usually, yeah. The P is usually more curved, like it's a hill instead of a peak, right? So, and then you have a T wave on that side. Yeah, a little one, and then a bigger. But, so you can look at that and you can see, okay, if you have a bigger decrease than you're supposed to, it'll indicate a depolarization event. It's not right. If you have parts of your heart that are electrically not working.
And for like heart attacks, like the thing that they kind of helps with, like to find out if somebody had actually having one and stuff. So like that's like normal like telemetry and stuff, kind of just like one beat and stuff. But what they kind of do, they get like a whole snapshot of the picture, it's like a 12-lead EKG, and it kind of shows like the picture of it, like EKG. Think of it kind of like wrapping around the heart. So like EKG is going to kind of show exactly like the conductivity like everywhere in the heart, so you kind of pinpoint like, all right, where's the function at like on the heart. And the thing they kind of look for is like this the ST elevation, which is going to be like right here. So like if this is elevated, it's showing that the heart's having like a bad like infarction.
So they 12 leads, what are the leads? The leads are things that they attach to the body and basically use it to surround the heart, right? So depending on where the lead is located on the body, what part of the heart it's closest to, which is the electrical signal from.
Yeah. And like the two types, like there's two types, like there's an instantly and instantly. The instantly is like whenever it's like you're having like a heart attack, but it's not like an ST elevation. But the STEMI is whenever you are having like ST elevation. And
That's
every,
what it looks like.
That's what it,
Acute one, like myocardial infarction.
Yeah.
How like they call it.
so you're not even seeing that as depression.
basically just don't.
See, I'm not an expert in reading EKGs. Like, I know the basics, but I'm not. I don't want to have to read one on a real person. I'm afraid I did it wrong. But yeah, it's really interesting. I love, like, I don't know, I want to study that more. All right, go ahead. I'm sorry.
So also it's related to lower incidences of a positive exercise test, which one of the like the diagnostics that we use on these type of patients is like put them to a stress test. Basically, they put them on a treadmill and with the leads on, and to see if the heart starts having some trouble, with electrical impulses and beats.
This lady, you can't put her on a stress test.
So now we know.
Stress test at all. She doesn't, she's no good at feeling.
Better have the crash cart next to her.
That's right, crash cart right there.
Yeah. So also lower incidence of heart failure, previous myocardial infarction, and dyskinesia at the ventricular lower. Dyskinesia is basically abnormalities, in the piece. Collateral circulation helps the patient with reducing ischemia, preserving ventricular function, and improving prognosis. What is atherosclerosis, and it's common among diabetics? Well, atherosclerosis is an inflammatory disease which initiates with cholesterol-containing low-density lipoproteins, which the low LDLs, the low-density ones, are the bad ones. The HDLs are the good ones. And a good number of LDLs in your blood should be less than 100. The good ones should be more than 50, more than 40 for men and more than 50 for women. Triglycerides should be less than 150. Diabetes is associated with changes in lipoproteins and plasma lipids in an atherogenic direction. Therefore, atherosclerosis is common among diabetics. If there is a physical examination, basically, most patients with diabetes, it always most likely come with blockages and calcifications. This patient developed diabetes at 32.
The problem with atherosclerosis too is that not only do you have that plaque, but if that plaque ruptures, it's going to start collecting red blood cells, right? And then red blood cells sticking onto red blood cells, that's a clot. So then that's what's going to lead to your heart attack.
Patient developed diabetes at 32. Is it typical for type 1 diabetes mellitus? Type 1 diabetes mellitus is known as the autoimmune diabetes, which basically means that your own body is killing its own B cells, the isolates in the pancreas. So its usual onset is during childhood and during adolescence, but it can develop later on in life, like you explained it to us.
It's like, have you ever noticed that some people develop like allergies as they get older? It's the same sort of thing. At any point during your life, your immune system can kind of go haywire. And so in this case, that's probably what happened with her. And she might have been diagnosed, she might have had it for a couple of years before she was actually diagnosed. So it might have been actually before 32, but still, it was probably like an adult onset. Was it like she always had not necessarily, no. There's a debate on, how much is gene related and how much is environmental.
Well, it said in one article, type 1 is not typically genetic, but type 2 has a genetic factor.
Right. So it's more likely environmental, but you probably have to be somewhat predisposed to that environmental because everybody's been exposed to that environment and they're not getting type 1 diabetes, right? So I think still under debate. Yeah. So she could have had like a bad instruction and then her immune system just kind of went, oh, don't like the pancreas, let's go. Yep.
It can happen.
Like people develop intolerance to lactose acid. A lot of stuff like that happens.
So it is not difficult, but it can happen.
So a test to kind of see if how like people how well regulated somebody's blood glucose level is like the hemoglobin A1C. So it's going to kind of give you like a 3-month picture of like if their sugar is going to like have been super high, have they've been really well controlled. And so that's like a level they can kind of like not really showing like the most recent picture, but kind of like overall like how well somebody's sugar is going to be controlled. And the reason why this is important is because like if somebody's sugar has been like uncontrolled for like a long period of time, it's going to do more damage to like their whole overall body, which typically like anything above 6 is kind of like a red flag.
So what is it actually testing? So, right, so if your sugar gets high enough, the sugar actually binds to hemoglobin in the red blood cells. This is why the tests don't good for the last 3 months. How long do red blood cells last? 3 months. So it's like the percent of red blood cells that have hemoglobin that are coated in sugar.
So it's a more definite diagnostic for that because if you go get your blood check and they just ate something, you should be high.
Right.
That's not, say that you have diabetes, but that one will because it's only one.
That's exactly. It also helps like doctors. It's like if you know you have an office where you're not taking blood. Yeah, you're gonna go 3 days and I'm not gonna eat my cake or whatever it is, right? So it's like a diagnostic tool for them just to like, keep track of their blood glucose. And for those of you who are going into the medical field, you're gonna see this. Patients lie all the time. So, not that you shouldn't trust your patients. You should. You should listen to your patients. But
And sometimes lying's not even on purpose, like,
it's not.
like,
Oh, no.
I'll take a personal example. I have arthritis, and I like to downplay my pain a lot because I don't want people to think I'm a baby because I'm chronically suffering. But the problem is because I do that, they just treat me differently because one, I'm fat, so they're like, oh, maybe it's fat or whatever. But second of all, because I downplay a lot of my symptoms, they don't think it's as severe, but they don't realize that I am somebody who suffers from chronic pain. Of course, my pain tolerance is higher.
I was showering an anesthesiologist after me, and like he had to ask the patient, he needed to know if they had smoked like weed recently, and of course they lied. I guess I knew they were lying. He's like, you have to tell me the truth. Like I'm not gonna get you in trouble with the law. Like I need to know this.
And I need to know,
And
like,
then they're
medically, this could kill
Well,
you.
I mean the other day. Like, yeah. How much?
The other day, every day for the last. One of the local family doctors here, obviously, we get a lot of diabetes and all that. And so every patient pretty much we were asking like, how's it been over the last however long? A lot of people don't actually check their blood sugar, which is really bad for them because you should know what it is. And so like they were like, yeah, it's been great. And like then their A1C would come back and it's like not great. Like,
Some funny stories about that is your patients they're asking themselves like, how can my sugar is so high? Like I'm only eating this little bit of food today, but then you put a blanket, like five Snicker bars, like
oh yeah, the refined sugar, and I had my Coke with lunch. Oh yeah. They go. Somebody, I was in the grocery store or something, and some people, you'll see this. Some people who can't read will stop you and say, "I can't read this box. I don't have my glasses." And so somebody asked me, "Can you read this cereal box or whatever?" And then they say, "So I have diabetes. Do you think there's a lot of sugar in there?" I'm like, "Oh my god, what is that?"
Like, you need some Wheaties, it's fine.
Too much sugar.
You need plain Wheaties.
No. I can't contribute to that. But yeah, they don't, they just don't, I don't know if they don't make the connection, I don't know.
I like
different options, but, yeah, go ahead.
So like if there's a life-threatening complication like diabetes, that's what I was kind of drawing out right there and stuff, is like ketoacidosis, which that's a formation of ketones. So like you start off with two, like we have excess acetyl-CoA. If you have excess acetyl-CoA, so 2 acetyl-CoA ended up becoming like acetyl-actyl-CoA, and then eventually becomes these 3 ketones. which these two are technically like the ketones because they actually have components, and this one's like the one which is the more stable one. But this goes into these two. This is secreted into the blood, which whenever this is secreted into the blood and it kind of breaks off, think of it like it's acidic, and in that going into your blood, it just keeps on dropping down the acidity, that's the reason why it becomes ketoacidosis. And your pH, which is 7.35 to 7.45, is pretty sensitive, and so if it just keeps on dropping, it can bring them to a coma and have all kind of other effects.
So if you're talking to a patient, you think it's going through DKA, that's what they call DKA. What do you smell in the breath?
A fruity,
Fruity.
yeah.
The acetone. The acetone has that fruity smell to it. And so it's one of the things like they're going through DKA. I mean, you know, like they're having problems. Yeah. And so why can't you use acetyl CoA in the citrate cycle?
Right there.
Like, why are you gonna go through and make ketone bodies? If I have acetyl CoA from the breakdown of fatty acids,
Because there's no oxaloacetate.
there's no oxaloacetate. It's a cyclic cycle. So you don't have other like starting point. You can't go through citric. But the body thinks it's in what kind of a mode?
It's starvation.
You're starving. But you're not. But it thinks that you're starving. Individual cells who are not getting sugar are like, I am starving. And so then you end up making all these ketone bodies, which are really not good for you. Not good for you, but better than dying, right? But your cells don't know that they're not narrow, but they sort of are. They're not.
Any questions about that? That was the easiest way I can break it down. So just going through the rest of the slides, we already kind of touched base on why I like elevated kinase levels. Like, what does it mean? It's not really having a broken heart during Valentine's Day, but it's just where your heart's having some muscle damage.
So why do we have creatine kinase? Like, what's the point of having creatine kinase in the heart? Like, what does it do? The kinase is phosphorylate. Okay, so it's phosphorylate. It will take an inorganic phosphate from ATP and attach it to creatine so that you have phosphocreatine. So basically, it gives you a reserve of inorganic phosphate so that you can, if you're a muscle and you use up your ATP, well, I don't have to go synthesize more ATP because I have this phospho group on the creatine. So then I can just pull it off and pop it back on ADP and I can have more ATP. So it's like another ATP reserve. Does that make sense? Yeah, it's like ATP storage. It's like a, yeah. But you're not storing ATP, you're just storing that high energy. No, it's just a one gallon. Oh, yeah. Oh, okay. Yeah. I'm sorry.
No, you're good. Are we short on time or are we just coming close? I was looking at. All right. So two medications that they use for people that are like antithrombolytics are like Coumadin and aspirin. They have the same goal, but the mechanisms are different. So Coumadin is a vitamin K antagonist, which is going to kind of it's going to inhibit an enzyme that is going to help make the coagulation factors, which are, I believe, I'm trying to think of something. 2, 7, 9, and 10.
Yes.
All right, cool.
Yeah.
And an aspirin is a non-selective cyclooxygenase inhibitor, which it inhibits COX-1 and 2, and those are needed to make prostaglandins, which those help make platelets. With platelets are the things that kind of form together to make a clot.
Right. So one is an anticoagulant. Coumadin is actually an anticoagulant, but the aspirin is actually an antiplatelet. So that's what we're asking that question. Yes.
Okay.
They both
I
prevent,
definitely misread the question.
they both prevent clots, but they do it in a different way. They target different parts of the clotting pathway.
Because it's coming to my brain.
Just achieve the same outcome, right? But they're not blood thinners, right?
Right.
Everybody's grandma calls them blood thinners, all the commercials call them blood thinners, but they're not.
And that's also why people have insulin take aspirin.
Right.
And a lot of people don't realize
So we talked about 5, and we really talked about 6. So yeah, so we can go with whatever. That was, what is elevated creatine kinase? We did that, we talked about that, right?
Yeah.
And we talked about
Insulin injections. The reason why they give it like in the sub-Q area away from your
oh yeah.
near your liver. So whenever you have like insulin that's kind of being made and stuff, it goes through the hepatic portal. So like your liver, you're not really going to get like all of it throughout your body, but whenever you give it sub-Q, you're going to kind of be able to have more access to the insulin that's going to be given.
Yeah, that's accurate. It's all about delivery. That's what so much of the project insulin lasts longer. Your body's just twice as drops. But insulin in general lasts. That's why like some people patient in general like once every 2 weeks or so, like insulin in their last.
Our photo takes away like percent of the endogenous insulin. But if you use another route, like for subcutaneous patient, you're gonna get most of it. It's not gonna go to.
It's not gonna get filtered out. Right. Exactly. Yeah.
So like, obviously, this is a very process and being like mentally exhausting for diabetics and patients. Like, what are some like therapeutic remedies or like hobbies that you think would do the control code of this?
Well, there's also life changes, so you can always exercise, reduce stress.
Definitely probably low impact for home health.
I mean, I should sometimes swimming.
I think of walking, swimming, maybe at night.
Or mall walkers. Yeah, mall walkers. Yeah.
It's about that says there's the portion is in the dosage. So if you take like consume a lot of carbohydrates and you don't break it down, then you're more exposed to
Yeah, too much of a good thing.
much
Bad.
better
Too much of anything is bad, right? We say this is toxic, that is toxic. Really, it's very good. So I hope this was cool. I hope this is really interesting. Something really different. And especially for y'all who are going to the medical field.
So this is not a senior seminar, like this.
Yeah, that's why I was like, I guess so.
Yeah, so this constant is on some of the sections on this stuff.
Like not just.
So spending time on it is good.
Understanding that it's crash. So, I'll have to.
Yeah, I gotta go.
It's fine.
So we gotta talk about which one is relative.
I wrote a tentative one down, but I sent that. For sure. Yeah, like I wrote a tentative one.
All right, cool beans.
Yeah, exactly. If you want to come see okay, I'm meeting on my own. Now we got to know. Oh, good. Oh, no, all right, there. Yeah, absolutely.
It can't be right next week.
It's not, oh, it could be next week. If it could be next week, go look at my calendar and book whatever you want. But if you want to do it in the next like 2 days, I gotta look. Fine. And go look at the calendar.
Probably write it all in one document.
Yeah. Oh, absolutely. I'm happy to do that. I'd love to do that.
Wait, let me end the Zoom because the Zoom is going.